Okay, so now it's my pleasure to introduce our next speaker, Dr. Will Debello. Dr. Debello is an associate professor of neurobiology, physiology, and behavior and a core faculty at the Center for Neuroscience. His research goal is to reconstruct the complete wiring diagram of local circuits to study the neural bases of information processing, plasticity, and learning. He studies the classic model of the barn-owl auditory localization pathway and uses an integrative approach combining cutting-edge techniques such as electrophysiology, auditory and visual receptive field mapping, immunohistochemistry, 3D visualization, and computer simulation to better understand how individual life experience is encoded by changes in wiring patterns. In doing so, Dr. Debello seeks to shed light on principles of neural processing that are employed across brain regions and across species. Today, Dr. Debello will share how he is using machine learning to unravel the brain's wiring diagram. Please join me in welcoming Dr. Debello.
Thanks a lot for that introduction. It's great to be here. I will be talking about the wiring diagram of neural circuits. And what I mean by that is the physical pattern of connections between neurons. Now, for the human brain, that's going to be a vast network, right? We have 86 billion neurons and 100 trillion plus connections. And the idea is that, all of our thoughts, our emotions, our memories, our skills are somehow constrained by this wiring diagram. However, only a small fraction of the brain's wiring has actually been mapped at the level of detail that I'm going to be talking about. So that's the grand challenge. Now, just to start off with some basic building blocks, we have on the right the brain, and on the left neurons, right? These are the cell types that are responsible for chemical and electrical signaling in the brain. Neurons have this really unusual shape. There's the cursor, where they have a cell body that has dendrites coming off it, often called the dendritic tree, because it looks like branches of a tree, and that's receiving signals from other neurons. And then that will eventually generate action potentials that propagate rapidly down these axons, which are the other parts of the neuron that are conveying information to the next neuron in the network. And at the tips of the axons are synapses, right? Those are the connections where a chemical transmitter is released by one cell and detected by another. Okay? So these are the basic nodes of just an immense network. And now the challenge is, can we actually map those connection patterns? Okay, the connectome is a word that was coined about 15 years ago by Olaf Sporns. It means the wiring diagram, or the complete set of connections in the brain, kind of analogous to the genome being the complete set of genes in the body. And Olaf Sporns noted at the time that there were several different spatial scales of mapping that were going to be relevant: the macroscale, mesoscale, and microscale. The first is the macroscale, which shown here is from an MRI image, magnetic resonance imaging, of a healthy human brain, and each of the wires are color-coded. Now, the wires there are actually bundles of axons, maybe tens of thousands of them together, right? So this is a broad spatial scale, or a coarse spatial scale, that shows which parts of the brain are talking to which other parts of the brain. Now, if you zoom in several orders of magnitude, we're down at the level of neural circuits. Shown here, there's maybe a few dozen neurons. Here would be a cell body. Here would be some of those dendrites. And you can see it's beginning to look like a very dense forest, right? And if you then go down a few more orders of magnitude, you get to the individual synaptic connections. So what's shown here are two dendrites in different colors from different neighboring neurons, and they're studded with these protrusions that are called dendritic spines, which are the sites of excitatory synaptic transmission in the brain. And we know this is an actual reconstruction of mammalian brain tissue that is based on imaging with an electron microscope. Okay? And one of the images that would come from an electron microscope is shown here. This is very high resolution. So we're seeing features that are nanometers in dimensions, right, which is 1/1000th of a meter. Okay? So this also points out the challenge then. We have features of the brain's wiring that are really important, individual synapses, okay, that require high-resolution imaging, but they're embedded in circuits that are, many orders of magnitude larger than that. So that presents this challenge, which is a multi-scale imaging problem. The human brain's 1,300 cubic centimeters in volume, but individual synapses are much smaller, that number there. So therefore, the techniques to reconstruct this network have to have both high resolution and high throughput capacity. An example of that, you already know, would be the Google Earth data set, right? So you can start, this is just a snapshot of Google Earth, where you start with a satellite level view and you can zoom into regional views, city views, and down to street level views. And then you can move over to some other part of the planet and zoom out again, right? So that is a multi-scale imaging data set that has both high resolution and high throughput capacity. Now, the technology for doing the analogous thing for the brain didn't exist 15 years ago, but advances in nanoimaging, computer science, and most recently artificial intelligence now make connectome reconstruction possible for small neural circuits. It's still estimated that a human brain is many thousands of years in the future with current technology, but the technology, of course, is changing. Okay, so that's the problem, a multi-scale imaging problem, and the techniques to do this relevant to brain processing are shown here. They start with an electron microscope, a scanning block face electron microscope in this case, where you have a bit of brain tissue that's been excised from a human during surgery or from experimental animals, as we'll see with my own work, and then you image the surface of that little bit of brain at high resolution, and you slice off the thinnest layer from the top of the tissue. You discard it, and you image the next block. And so what you acquire is this stack of images, okay? And this is the data set. That's our petabyte scale data sets. And in fact, who better show that than the people who brought us Google Earth? Her link is not working. Here we go. A browsable petascale, that's petabyte scale, reconstruction of human cortex. This is a project, it's a collaboration between Google and Harvard, the Jeff Lichtman Lab. And there's many very cool links here, and I was just going to show you one of them to give you a flavor of the type of data. Okay, so on the right is the reconstruction that was accomplished by Google, and on the left is the raw data. So these are that series of image stacks that I was just talking about that has very high resolution. Put my glasses on here. And what we can see is we can move between these and identify the structures in the reconstruction and their source EM images. In fact, I can zoom in on this, and you can see here's the cell body that's lighting up when I mouse over it. Here's an interesting structure called whorl synapses, okay, that are lighting up when I mouse over it. This is the primary data. And what's important here is that it is a three-dimensional data set. Shown this way. So now I'm panning through it, and you can see the features changing, right? Because it's a series of two-dimensional images that are stacked. So you can get a sense of how much data is here, right? This is a huge data set. And to go from the raw images on the left to the reconstruction on the right involves annotation, okay? You have to zoom in to the synapses, the little bits of dendrite and axons and cell bodies, and actually sketch the contours or trace the contours. And then put those all together in a giant jigsaw puzzle to get these reconstructions, which are the wiring diagram. Okay, so that's the challenge here. We have this immense data set that requires something like human visual capabilities to identify the features of interest, but really ideally would require, a supercomputer speed to achieve. So that is what I'm going to come back to at the end of the talk, is how machine learning is actually accelerating these reconstruction pipelines as we go from the images on the left to the reconstructions on the right. Okay. Here we go. So now, into my lab's research. As mentioned, I use the barn owl auditory localization pathway as a model system. So neuroscience has a great tradition of using animal models for what they're good at, right? A species that has some unique capability that's evolved over evolutionary timescales. We're interested in the neural processing that underlies that ability. Barn owls, as nocturnal hunters, rely on sound localization for survival, and so the hearing, their auditory system, is very sophisticated. And that's the purpose of this model that's been studied for more than 40 years, is to be able to ask questions about how the auditory system processes information and how it learns. And those questions that we typically ask are not specific to owl, but rather relate to common principles that operate across circuits and across species. One paradigm that's been very useful for the lab over the years is a learning paradigm here called prism adaptation. So yes, this is a barn owl wearing prism glasses. The glasses displace the visual field to one side, and what this does is create a mismatch between where they see things and where they hear things. The brain has to solve that, and it does through rewiring. So we've known this for a while. These paradigms, I should say, this was invented by Eric Knudsen, and the owl system was pioneered by Mark Konishi. So these systems have been around for a while, and we've known that rewiring is necessary to drive adaptation to prisms, but we only recently now have the techniques to look at the details of that rewiring. So we obtained electron microscope image stacks like the one I showed you, and we set a team of undergraduates to reconstruct the neurons and the synapses, and this is what they came up with. This is a space-specific neuron outlined in green, and it's studied with this unusual type of dendritic spine that we call toric spines for their holes, toroid. What's more significant is that the toric spines receive multiple synapses. So each one of these balls is a synapse coming in. And in fact, the toric spines can receive up to 50 synapses, which means they're structured to act as integrators. This is a novel role for dendritic spines, which typically have one synaptic input. So we're very interested in this convergence of information towards toric spine. So what I'm going to show is the single toric spine here, disembodied from this neuron, and now with the complete set of synaptic inputs that were reconstructed from the EM data sets. And it's shown here. It's a pile of spaghetti. It's hard to really see anything. The toric spine is buried within. It turns out that there are 61 axons coming from other neurons that are infiltrating this toric spine, only some of which are synapsing with it. Okay, so think about that. All of these axons, which are in different colors, are providing potential inputs to this integrative structure. Okay, but only a fraction are actually integrating with that. That has really important implications for plasticity and learning that are shown here. So the connection fraction, very simple formula here, is defined as the number of axons that synapse with the spine divided by the total number of axons that are within easy reach of that spine, okay, or potential partners, right? So there's 10 axons synapsing with the spine I just showed, but 61 potential partners. There's 51 other axons not synapsing with it. So it has a low connection fraction, actually just like mammalian cortices. And here's the key thing. What this implies is an enormous capacity for plasticity. Okay? Because if you imagine the spine could be sampling the different potential partners, how many different wiring diagrams are possible? It's 61 choose 10, or 90 billion. wiring diagrams just for this one spine on one neuron, right? So if you extrapolate out to whole neural circuits, or imagine even the human brain, it's a virtually infinite number of wiring diagrams. And the fundamental idea driving this field is that at least some of our individuality is stored in these different patterns of connections. Okay, so on to machine learning. Machine learning applied to connectome reconstruction. So the idea is to merge human-like pattern recognition, looking at those EM images, operating at supercomputer speed. That would lead to an accelerated reconstruction pipeline. Reconstruction has up until now been by far the rate-limiting step. You can generate petabyte-scale image data sets in a matter of weeks, and they would take 10 years to analyze. So this is important. So the way to do this, we need machine learning algorithms. We also need training data from human annotators who are able to go through and make very dense reconstructions of small volumes of tissue and use that to train the machine learning algorithm. And that's shown here. Here's an example of the training data. This is also called ground truth. It's what the humans are feeding the machine to tell it this is what you want to recognize, as we've seen in previous talks. So the machine learning algorithm learns what synapses, axons, and dendrites look like, and it can then identify these features over much larger volumes in much less time, right? So that's what we've done. We've used that training data to train an artificial neural network, which is shown here as the black box. And that artificial network can now take raw EM images and produce contours from them. So this is the beginnings of assembling the wiring diagram. In fact, the tracing of those individual contours was the rate-limiting step previously. So how does it do this? It uses generative adversarial networks. They cycle image cross-currently to create a pairwise alignment that can be passed to a least squares. So I can explain that, but actually it's kind of magic. So I can't explain how the machine learning algorithm actually works. And if I tried, my explanation would be something like this, right? Very haphazard. These algorithms were written by Matthew Medani, who's at University of California at San Diego, working with Mark Ellisman, who's the director of the National Center for Molecular Imaging Research. So they are the gurus who put this together. We have applied it to the owl tissue. Here's the output of the machine learning algorithm. It works very nicely. It identifies synaptic vesicles, which I haven't yet talked about, postsynaptic densities, their mitochondria as well. Most important, it is producing what's called a boundary mass. So it's actually identified all the different pieces of neuropil, that's the axons, dendrites, and synapses that are just in that dense forest, right? And it's picking those out, so now it's effectively tracing them. So now what the human does is come in with this circular tool and just click a button and say, I want this one or I want that one. Okay? And this leads to very rapid reconstruction. And as just one example of this, here's another pile of spaghetti, a dense reconstruction from the owl brain that was accomplished by one human annotator in 2 weeks. This previously would have taken a team of annotators months to produce. So this is the practical benefit of the machine learning. Okay, that just leads me back to where we started. In summary, the connectome, aka the brain's wiring diagram, we believe is a major storage site of our individuality, our unique memories and skills and thoughts. Charting these connectomes at the microscale was just previously impossible, from a time and labor perspective, but machine learning is really promising to accelerate that. And in analogy to the Human Genome Project, which started in 1990 and took 10 years for the first human genome to be sequenced, it will take a while to do the first, circuits, but as technology continues, there's a reasonable expectation that we will actually begin to map circuits across many different brains and regions and species, including still probably a whole human brain is in the future. That's it, and I'm happy to take questions.
mentioned the ratio of 61/10
Yeah.
for the axons and the potential partners. I didn't quite understand. One, why are the potential partners there, and whether they are wired. I think you said they're wired as well.
Yes.
So thank
Yeah,
you.
that's a great question. So why are the potential partners there if they're not actually synapsing with it? It's a great question. The potential partners, all of those axons that are there, are carrying information that might be useful to the neuron. And what happens is that as they come in, they're providing potential input sources to that neuron. And the neuron, on the basis of experience, is deciding or selecting which it's going to synapse with. Now, the key thing is that your experience can change, right? And that it now might be appropriate for that neuron to be sampling a different subset of those potential inputs.
So if a species or human being or barn owl has limited experiences, it limits their plasticity.
Yeah, absolutely. That's a really well-established finding of developmental neuroscience in general, that during especially early development, there are critical periods and then sensitive periods for circuit development where the brain, for a critical period, absolutely needs normally patterned experience to develop properly. And if the brain does not receive that when you get past the critical period, even if you restore normal patterns of experience, you're not going to get a normal pattern of connections. The related concept of the sensitive period, which is kind of a continuum of the impact of experience on circuit development. Now, with that said, there are ways to get around critical periods and sensitive periods. I'll give one example from an owl. It is a sensitive period, not a critical period, but owls adapt to prisms only as juveniles. Once they hit sexual maturity, if you slap prisms on, they can't do it. Except there are two ways to get around that. One is to use what's called incremental plasticity. I didn't mention, but the prisms displace the visual field by 23 degrees. Naive adult owls cannot adapt to a 23-degree prismatic displacement. They can adapt to 6 degrees, and once adapted to 6 degrees, you can put another 6 degrees on, and you can step them out. This is a principle called incremental plasticity that's actually been put into practice for at least 20 years by a company called Scientific Learning that's produced a software called Fast Forward, which is a remediation for auditory processing deficits in kids. I won't describe the details of that, but it uses the principle of incremental plasticity. Another way you can get naive adult owls to adapt to prisms is provide them great incentive, which is what they do, what they want to do, what they evolved to do, which is to hunt mice. And so if you actually give them live mice and require them to hunt in a little pen about the size of this stage, it's that an engagement or arousal, it's probably called arousal, is enough to open their brain again to be receptive.
Thank you very much. I'm very far from this area, so sorry if my question is silly. I'm just curious, you speak about wiring and rewiring. What is the mechanism of wiring and rewiring, shifting all those connections?
Yeah, great question. Up until 2001 or 2002, the overarching paradigm in neuroscience was that after development into young adulthood, your wiring diagram's set. And the plasticity after that is largely driven by changes in synaptic strength, something called LTP, long-term potentiation or long-term depression. Those for sure operate. Just at the turn of the century, there were techniques invented, not these, to actually look into living brains of mice and to visualize the dendritic spines over a period of weeks or months. And it was shown that the spines actually extend and retract. They're motile, okay? They're not fixed. And it was later shown that a fraction of them that extend and are stabilized are forming a new synapse. So in fact, there is this capacity, and that continues into the adult, a lifelong capacity for what I call microanatomical rearrangements, which are just those dendritic spines. Here's these dendritic spines. These guys right here, these are dendritic spines, are sampling their local environment for new synaptic partners. So that is the presumed mechanism. Great questions. I'll stop there. I think I'm over time, so thanks.
Okay, so thank you, Dr. Debello. Thank you, Dr. Moxon and Dr. Hanks. Really awesome talks. I want to do a little bit of business, and then we're going to take a break, and we're going to come back. So I wanted to just let you know that right outside of these doors, you probably saw several beautiful NeuroArt images. So these NeuroArt images came from our research labs. And the brain is really not only remarkably complex and has incredible capacity to allow us to do what we do, but it's also gorgeous. And you've gotten a little bit of sense of that through some of the talks that we've seen. In 2020, the Center for Neuroscience launched the NeuroArt competition to secure beautiful images for our lobby. This year, we've received 33 entries, and today we're presenting the 8 finalists to you. We need your help now to vote for the top 3 favorite images, listing 1 through 3. We have cash prizes of $300 for first place, $200 for second place, and $100 for third place. And these are going to be presented to the contributors, and the winning images are going to be added to the gallery at the Center for Neuroscience after this event. So please join the fun and cast your vote by 5 p.m. today by collecting a ballot and dropping it in the ballot box. We have a volunteer that's outside. She can help you figure out how to do that and what to look at. And I think we're going to be announcing those winners next week. Yep. Okay, so if you are interested in visiting the Center for Neuroscience to see our NeuroArt gallery, we would be really happy to host you. Please sign up at the information booth, and we can arrange a time for a tour and a gallery viewing. Also, don't want you to forget about the neuro activities and the neuro posters. We have, again, 12 of those interactive booths and 6 neuro posters with our great graduate students that are going to tell you all about their awesome work. If you have volunteered to be a neuro poster judge, please be sure to complete your visits during this next break by 3:45. And so that we can collect your rankings, and the winners are actually going to be announced at the end of the program today. What we're going to do now is we're going to take a 40-minute break to give you some time to process all of the amazing information that you've just received, to see some of those booths, to vote for your posters and your NeuroArt, and we hope that you'll come back by 3:40 for the additional talks. We've got 2 absolutely outstanding talks, so don't miss those. Okay, enjoy your break.